So I would like to speak about honeybee pheromones, putting the accent about some new discoveries or some new unsuspected roles that we found for pheromones, or for some pheromones at least, in the last time. I'd like first to start to telling you a little bit about how pheromones were first discovered. And actually the main lesson is that, well, you know very well that pheromones is a widespread concept that you will find practically in all living animals. But the source of the discovery of the concept of pheromone was a little insect. So you see, insects are extremely important for research, for the discovery of relevant concepts in biology. So this goes back to a long time ago to history, and the first one who reported actually something that might be inspiring for the concept of pheromone was the French naturalist and entomologist Jean-Henri Fabre, which you can see, oh, well, there's no laser, you can see here. And he was actually collecting insects in nature everywhere. He had a house in the countryside, and he went actually to a forest close to his house actually, and he collected a female butterfly of the giant peacock moth, which you can see there, by the way. And he brought it to his house, and well, the legend says actually, this is what you see in the cartoon below, that he was totally surprised by seeing lots of males coming far away from the forest actually to fly around the window where he had put his female moth within a cage. So you see here the female would be here in the cage. That is Fabre, and all the males are coming around to the surprise of the people in the village. So, and he started doing some experiments actually, so he assumed it could not be visual, how the moths would be, the males would be a female from the distance. So, so he decided to put a glass jar on top of the female, and then the attraction stopped. So at that time, he concluded that, without knowing anything about pheromones or how it worked, that there should be an olfactory signal released by the female, and that should be the basis for the attraction. That stayed there for many, many years. Nobody continued this kind of investigation. So you see, it's 1925 or something like that. And many years later, actually, in the 50s, or even a little bit earlier, actually, we had this very important German scientist, Dietrich Schneider, actually, who decided to go back to these kinds of observations and study the basis of these attractions. Dietrich Schneider actually decided to focus on another kind of moth, which is the silk moth, Bombyx mori, which you can see here. Actually, you see here the male. You see here the male, and this is the female. There is an important difference when you look at the antennae of these two guys. Actually, males have huge ramified antennae, you can see here. And females have also, obviously, antennae, but they are smaller. They have not this kind of spectacular structure that males have. So one of the obvious conclusions is that, okay, males might be detecting some kind of scent emitted by the female, and this is why they have these incredibly huge antennae, because, as you might know, the antennae are the nose of insects, okay? So he actually realized also that these females, at some point in the moment of the sexual reproduction, they evaginate the posterior part of the abdomen and expose a gland that is normally within the abdomen, which you can see here. And that's the pheromone-producing gland in the abdomen of the female Bombyx mori. So the question is, why did he decide to work on Bombyx mori, on the silk moth? For a very simple and practical reason. I mean, you can see these guys here, and you can see here, for instance, a male. You recognize it because of the huge antennae. And you can see the female. And you see something very peculiar in these moths. They are like, actually, I would say, white mice in a laboratory. What I mean by that, these are fully artificial animals in the sense that they have been selected by humans through thousands and thousands of generations to produce silk. And they were selected, and the result is what you can see here. These guys have huge bodies and small wings. They cannot fly. They have lost the capacity to fly. So for the scientists, because then they stay in the containers where people raise them to get the silk, the cocoons and the silk and so on. So they cannot fly. So it was extremely practical for Schneider and the other scientists to use these animals because they could track them just on a table and see how is the orientation behavior instead of trying to figure the flights and reconstruct the flight, which is almost impossible. So, and this is actually what they did. As I say, they have lost the ability to fly, and then it was very simple to create a lab assay actually to study this sexual attraction. And this is what you see here. This is the male on a table, and on the other side, this is the female. This is a real video recording made from Schneider group long ago. And here you see the female, and he has a little ventilator here just sending the air to the male. And this is the reaction of the male when it gets the signal, the odor coming to him. So you see it turns towards the direction of the female and then goes there immediately, male, female. So you see the ventilator is sending the signal, and the male is going towards the source of the orientation. This is something like what Fabre observed at the very beginning. But here it's fully controlled. You can see absolutely everything, all the sequences of this attraction. And this is the moment, of course, of the sexual reproduction. And then comes another experiment they did, which is to show that the basis of this attraction is indeed in the products released by this abdominal gland you already saw in a picture. So now you will see that the experimenter will take a female from a little petri dish here, and the experimenter will gently press the abdomen just to make the gland go out, and then he will just put the secretion there, you see? And now the ventilator is activated, and here's the effect. Here it goes. Poor guy, actually. But he believes it's true. And so he arrives there, as you can see here, and now the frustration is complete because you can see it's turning around, but there's nothing there. But so this was one of the many experiments done by Schneider to show that indeed there was something there produced by the female attracting the male. Then, of course, he affiliated or he collaborated with Adolf Butenandt, who was a very famous German scientist. He got the Nobel Prize in 1939. And they worked together actually to identify the basis or the chemical basis of these pheromones. At that time, nobody knew anything about this kind of concept, pheromonal concept, and so on. So this is just, you can see these are cocoons of Bombyx mori, and this is Schneider. Actually, this picture was made just to show that they had to sacrifice 500,000 female butterflies, actually, to obtain only 6.4 milligrams of the pure substance of the pheromone. That was actually insane, actually, but now we have very different sophisticated methods. You would not need to do this. With a single female, you could extract the pheromone or the components and so on. But at that time, of course, it was difficult and different. And this was the discovery. They found actually that the pheromone was made of two molecules. First one was first discovered, which was called, it's an alcohol, that's a group alcohol. And it was called bombicol, from alcohol, and from Bombyx mori, bombicol. And then years later, they discovered that there was another component in the pheromone, an aldehyde, so which they call bombical, from aldehyde. So actually there are two components, an alcohol, which is very volatile and attracts from distance, and bombical, the aldehyde, which is perceived from closer ranges, and it's actually guiding the final detection, the final approach. And from there, actually, the same year, you see that they identified the first molecule, Carlson and Lucier proposed for the very first time the term pheromone. So, okay, so until Schneider and Butenandt didn't do these works, nobody used the term. It was inspired by their work. So, and the definition that you all probably heard at some point is that pheromones are chemical substances which are released outwards by an individual and which act as chemical messengers, so within a species. So it's species specific, so it's for a member of my same species. And they trigger specific behaviors in relation to the message being transmitted. So there are pheromones for sexual attraction, that's what we saw. There are pheromones to mark your territory. There are pheromones for attack, aggression, things like that. There are many, a broad spectrum of possibilities, but it works within species. And they release specific innate behaviors, like the attraction you saw from the male. Pheromone is there, innate attraction to the source of the signal. So another important thing to keep in mind, which you probably learned from the example of bombyx, the silk moth, is that contrary to what many people think, pheromones are not one molecule, okay? You saw the pheromone of bombyx was made of 2 molecules. Well, pheromones are rarely one molecule. You might have cases where you have one pheromone, one molecule. Indeed, in the honeybee, we will see one case. But most of the cases, I would say 90% of the cases, pheromones are integrated by many molecules. In fact, they are kind of bouquet of molecules. And this is important because the information is contained not only in the composition, in the molecules, but also in the proportions. Some species might have exactly the same molecules as the other, but the proportion may change from one species to the other, and then that gives the specificity of the signal for species A and for species B. So keep that in mind, because in the examples that we will see that many of these pheromones are made from many molecules. Okay, so, and of course, we all know that mastering this concept of pheromones and their composition and so on, it's very important because these pheromones are playing a very important role in the biological control of insect pests. So pheromone traps have been developed for many species which are pests and so on in agriculture and so on. And of course, given what we discussed yesterday, you probably will agree with me that they would be a preferable solution to the use of pesticides. But of course, it's not easy actually to find the full composition of a pheromone and to conceive then an industrial or economical solution to apply it in the field. There are lots of steps to do before reaching that. And this is why actually, at least in France, we are still waiting for a pheromone for capturing Asian hornets, you know, which is a very important problem in France. But I hope it will come at some point. So we will speak now about bees. After having this introduction with some important concepts of, about pheromones, let's speak now about bees. So bees are a very interesting case because they are a social organism, not like the silk moth. And we know very well, you know very well, that pheromones are an extremely important basis for this social organization. It relies to a large extent on the presence of many different pheromones. And actually, the honeybee is one of the species for which the pheromones, the different spectrum of pheromones, has been better or best investigated or in more detail. And I think that many people have contributed to that, but being here, of course, it's important to give tribute to at least these 2 persons. There might be other more working with them, of course, at that time, but John Free and Colin Butler actually were extremely influential in the characterization of honeybee pheromones at the time they were in Rodamstedt, if I'm correct. Yes, so so we have in the honeybee actually multiple pheromone, so which means multiple kinds of messages, different messages, which are released by multiple senders, so different categories of senders. I'm just giving you here a short overview, and then the idea is that I will go step by step over these categories to provide more detail about these. So of course we know that there is queen pheromone, which is responsible for social dominance, cohesion, sexual attraction, and so on. We know also that there are brood pheromones, which are released by the brood at different stages of their life. We will see that, which promote foraging, nutrient supply, cell capping, things that we will see later. we are also having among these multiple messages and multiple senders guards releasing pheromones that are responsible or that activate some different behaviors related to the defense of the colony, to attacks, and so on. And also we have pheromones released by foragers themselves, that you can see here, which might be used for marking attractive spots like the hive entrance, like food sources, especially those which have not a prominent odor, and so on and so on. We will go through these actually in the next slides. So let me start actually by showing you how rich is the pheromone world or possibility in terms of a honeybee body. So this is a silhouette, actually the legs are not there, but of a honeybee actually, and here you see a series of glands that are indicated which might be related to the production of pheromones. So we have, for instance, mandibular glands which in workers produce 2-eptanone, a pheromone that is associated to repellency and rejection of intruders, among others. So, and you have here the maxillary glands, which we will see actually are related to other kind of compound production. The sting, which is related to the Koshevnikov glands and other glands here. So you see there's a whole spectrum of possibilities. Important thing is that these glands should communicate with the external world via some ducts and so on. So in order to the pheromones to go to the environment and pass the message they need to pass. So let's speak about queens actually. I will be brief here, but actually this is actually a very important work done by Colin Butler actually, and others came after him obviously, but the queen pheromone is having multiple effects, and we know that there is, it's made of 5 essential components. It's a bouquet again. Another example where you see pheromone is not just one molecule, but a group of molecules. And these pheromones actually of the queen, which you can see here, is made of 5. You see 4 actually, because one of them has 2 variants. Cis and trans is just a chemical term to indicate that this could be with this in this way or with this in this way. But so if you consider this variation in this molecule, you have 5. So one of them is the oxodecenoic acid that you can see here. This one. This one is the hydroxydecenoic acid with the 2 variants. And then you have these 2 HOB and HVA, which is the methyl hydroxybenzoate and hydroxymethoxyphenylethanol. Very complicated, so not important. I think if you just call them HOB, HVA, 9-HDA or 9-ODA, that's enough. So this compound of molecules, they are very relevant to control different aspects of the social life within the hive. So, for instance, these queen pheromones might actually intervene in swarm clustering, in the sexual reproduction context, so attracting drones during the nuptial flight, in promoting also the returning behavior that you see within the hive, and many other aspects. So these green arrows mean activation, actually, and this means the red arrows means inhibition. So actually, they might also, of course, inhibit worker reproduction, ovary development, and things like that. Not all the compounds are doing exactly the same. Some compounds might be more important for some of these functions and other for other functions. For instance, when we speak about ovary inhibition in the workers, it turns out that 9-ODA and 9-HDA are playing a more important role than the 2 others for this component. But in other behaviors, you might see different effects. Important thing is that they have to be there together to work in a synergistic way to ensure all these different functions. Because as you can see, all these functions are essential for the social organization and the social cohesion of the hive. So, but these are not the only pheromones. There are other pheromones that the queens release that have been less explored, and I think that it should be more work on those, because we know less more about these 2, for instance. They are redundant in the sense that they contribute to similar functions as the one we have just seen. And that's good, actually. In biology, redundancy is good, which means that you have a function ensured not only via one channel, but also by another different channel, you get to the same endpoint, which is good because if one fails, then you have the other always available. So, for instance, long ago, Lenski and Slabeski identified or characterized the so-called tarsal footprint pheromone. So, in the tarsi here, you have some glands that in the case of queens release some kind of oily secretion. So it's not so volatile actually, but it's there, and the chemical composition has been actually characterized. The names are not important. You have a series of alkanes, alcohols, acids, esters, and so on. And actually it was shown that these tarsal secretions are very important to inhibit queen cap construction. And this is actually the explanation why these queen caps are built in the border actually of the frames, because the argument is that, and they show that by a series of manipulations, is that when the colony is extremely crowded actually, queens do not reach the place where the queen caps are built. And because they do not reach the place, there is no tarsal secretion on there, which promotes there the construction of this kind of queen caps at this particular level. And there's another kind of pheromone that is less explored, because what I like to say is that, of course, we know this about the tarsal one, but since then there has been not much work about this. So more work, in my opinion, should be done in this direction. Similar situation for the pterygal gland pheromone. So long ago it was discovered that here at the dorsal level of the abdomen you have some glands, the pterygal glands actually, that release a series of compounds, very complex blend. You have esters, alkenes, carboxylic acids, and so on, among others. And so a recent work, actually, very nice work, very, very new, actually you see here by Tom Wenzelers. He actually wanted to know what these kind of substances are doing, and he was able to show that by playing, by doing different kinds of blends and so on, having all the molecules identified, that what this pterygal pheromone is doing, it's also inhibiting actually the development of the ovaries in the worker. So you see, the mandibular pheromone of the queen mandibular pheromone, was doing exactly that. The pterygal gland is also doing the same. Okay, so and he showed that this pterygal pheromone was as efficient as the mandibular pheromone to stop workers from reproducing, from develop ovaries. So again, that's another example of redundancy. You see, queen mandibular pheromone is doing this inhibition of ovaries, the pterygal gland pheromone is doing the same. They both work together, and you have this synergy of effects, which ensures that the thing will work properly within the colony. Let's speak now about brood. So we leave the queen aside for a moment, and then we will speak about brood. The case of brood is always something that is fascinating to me, because we have here some kind of non-finished organism, okay, the brood, which is sending signals which control somehow the working of a society. It's like a crazy if you think, well, what's going on, okay? And these brood pheromones, there are two, in fact. There are two kinds of brood pheromones. Both were discovered or mostly studied by Yves Lecomte, who is a researcher in the south of France, in Avignon. But these discoveries he did when he was a postdoc in USA working with Gene Robinson in Illinois. So the first brood pheromone, which was identified, which is called the steric brood pheromone. You see here the term steric because it's related to the chemical composition it has. It's a complex blend, again and again, you see, a complex blend made of 10 fatty acid esters. This is why it's the steric brood pheromone. And this is found mostly in older brood, okay? So here you have the names of the molecules, non-important, very complex. But this pheromone was actually characterized by Yves, and he found that it inhibits the ovaries again and again, not only the queen. You can see, here you can see ovary development, that he measured, and he has different groups. So he had here a control group, and he has here a certain amount of pheromone. You see, this is a small quantity of pheromone. This is a more important quantity of pheromone, and this is an even more important quantity of pheromone. And you see here with the smaller quantity, there is a small decrease, again, no significant. But when he add a little bit more, you see there is a reduction in ovary development induced by this brood pheromone. So again, it's kind of crazy because you can see that it's not only the queen, it's also the brood that can produce the same effect. Okay. But it's not on, I mean, this brood pheromone is doing more things, not just inhibit the development of worker ovaries, as shown by this graph, but it also promotes foraging, in particular pollen foraging. It promotes larval cell cleaning, and it promotes also cell capping. All these effects were demonstrated by the research of Yves Leconte. To my knowledge, at some point, there was a company selling commercially this pheromone, but I have the impression that someone told me that they stopped doing that. I don't know why. But you might know better than me in terms of commercial applications. I was telling you that there are two pheromones, two brood pheromones. First one, the steric one, the one we just saw, made of this complex blend. And this is the second one. And here you have one of the rare cases in which one pheromone, one molecule. Remember, I told you that that might happen, but it's kind of not common situation. Well, this is the case. So here, this was discovered also by Yves Leconte, but later. And he discovered this single molecule called beta-ocimine. You can see here, beta-ocimine. And he found it mostly in young brood, okay, while the other one was mostly released by older brood. And he showed that this one is a very strong promoter of foraging activity. The other one was also able to promote foraging activity, but this one was much better in that direction. So it accelerates the maturation of the workers so that they become foragers earlier than expected. That he showed, for instance, in this experiment. This experiment was done along 2 consecutive years, 2008, 2009, and the results are essentially the same. So what you see here is the control group. So these are colonies that were not exposed to some artificial beta-ocimine or experimental delivery of beta-ocimine. And you can see here that this is the average age of first foraging. So these guys are living at 34 days when they are 34 days old, okay? Here in 2009, of course, the situation changed, probably because there were more brood or there was more need for resources. So the reference points here are different. So here they started at when they were 1 month old, and here they started when they were 20-something days old. But you can see what happens if you add the intermediate or the high dose in the pheromone. In both cases, tack, you decrease the age. Okay? So meaning by that when you add, say, an intermediate dose, okay, they start foraging earlier, not at 34 days old, but at 30 days old, or even at 28 days old. And same here. You see, there is a decrease. So you can see that this beta-ocimine, it's a very interesting pheromone because it shows that the brood are able to control the foraging activity of the whole colony, which is, again, for me, kind of crazy situation because this is like unborn individuals controlling what the adults are doing, okay? So, and it also inhibits the ovaries of workers, okay? So, again, but the main factor, one of the main factors is the promotion of foraging. So let me show you some of the things we also show recently in a group. I mean, in our group, we have Gabriela de Brito Sanchez, who is a researcher who is focusing 100% on the regulation of feeding activity in bees. So what are the mechanisms that regulate appetitive motivation? What makes a bee interested in going for food and things like that? And he had the idea that beta-ocimine might be actually affecting decisions at the foraging site. What we mentioned so far is that beta-ocimine was driving the bees to go out for food, okay? So what she wanted to know is that what happens once they reach the food place. Is the beta-ocimene also changing the way they deal with food? And so with Rafael Carvalho, who is a postdoc in the team, they did the following experiment, which is very funny. So they had two groups of bees. They decided to have hungry bees, of course, it's a normal situation for a foraging experiment. But then they had semi-satiated bees, so they really fed the bees with a lot of food. They had not 100% full crop, but these guys were quite full, actually. And then they had these two groups, and then they exposed them to two situations. One group was exposed to beta-ocimene, the one we just saw, and the other group is exposed to something called mineral oil, which is just something without any odor. It's just a control, okay? So they had control and beta-ocimene. And then they placed the bees in a little syringe, like you can see here, and they had a little tip full with sucrose solution here. So then they allow them to stay for 1 hour within this syringe, and then at the end of this hour, they measure the consumption of sucrose solution to see whether, for instance, beta-ocimene not only triggers flying for food, but also changes food consumption once they are at the food source. And so this is the result they obtain. So this is consumption in microliters, and you have here the semi-satiated bees, and here the hungry bees. Obviously, as you would expect, the hungry bees eat more, okay? It's normal, they are hungry. So each little dot is a bee, is the consumption of a bee. This is the average value. So you see that in average, the hungry bees actually fill their crop, 60 microliters or something like that. And the other ones consume a little bit, but not too much because they were semi-satiated. So these are guys that were exposed to mineral oil, so no odor, just control. What happened now to these two groups, semi-satiated and hungry, when they were exposed before being in the syringe to beta-ocimene? This is what happened. So the semi-satiated, when you have a small percentage, a small dose, nevertheless, they increase. And if the dose is higher, they increase even more. You can see here. What happens to the hungry bees? Same situation. Even if they seem to have consumed a lot, they can still consume more because this is the result of beta-ocimene exposure. By being exposed to a small dose or a high dose, consumption increase. So this result is interesting because it shows that beta-ocimene not only promotes foraging, but also somehow changes the decisions once they are at the food source. So actually what beta-ocimene is doing is changing the decision of consuming less or more, so that at the end bees return to the colony with more food after having been exposed to beta-ocimene. Well, the results for the semi-satiated bees you saw already, and the same for the hungry bees. But that's the essential message. So bees return to the colony with more food, okay? So, so these brood pheromones, actually, they act together at different levels and different scales. Remember, we have the steric pheromone made of this blend of compounds, and we have the beta-ocimene. And I mentioned that young brood is mostly releasing beta-ocimene, while old brood is mostly releasing the steric pheromone. So we have a situation like this, actually. So young brood, actually, so here you have the 3 different kinds of colors and arrows. So the black arrow indicates real brood, what real brood is doing. The red arrows indicate what the steric pheromone is doing, and the blue arrow indicates what beta-ocimene is doing. So for young brood, you see lots of blue arrows, actually, because this is beta-ocimene working at this stage. You have some kind of small arrow for the steric pheromone because it's really not acting fully at this level. But once you actually move to this old brood stage, then you have the steric pheromone everywhere and not the blue arrows, not the beta-ocimene working, okay? And then you see the different effects we mentioned: delay behavior maturation, trigger cell capping, over-inhibition, which you reach by the 2 pheromones, and so on. This is actually the summary that we can that I would like to provide to you about these brute pheromones. There's a lot of things to understand yet there. I mean, there are more and more questions. So, for instance, an obvious question I'm not asking, may I remind you actually that I am a neurobiologist, as we say yesterday. So the essential question to me is, okay, they smell these forager works, they perceive these brute pheromone, either one or the other. What happens in the brain? What is changed actually for them not only to go out, but to decide to eat more, as the case of beta-ocimene. We're working on that right now, actually. We have some experiments going. We might discuss that at the end during the questions, but I like to move on first because otherwise there will be probably no time. So, as I say, it's just to say that how many questions actually are there, it's crazy. I mean, it's no-win situation, and that's great, actually. So let's speak about what guards are doing. So you have here the well-known fact that guards actually release alarm and defense pheromones, mostly to respond to aversive situations or to actually aversive stimuli. And I like to mention here, actually, I will mention essentially two kinds of pheromone. The ones released by the Koshevnikov gland, which is the stinger pheromone, actually here. Again, it's a bouquet of substance, we will see that, which triggers the coordinate response, the attack response towards enemies or dangers and so on. And the other one, which is a pheromone released by the mandibular glands. And then this is another case where one pheromone is one molecule. So, which is the fact that these mandibular glands in the workers, in the guards, release 2-heptanone, which is, again, a single molecule, but which very, very powerful effects in terms of repellency, defense, and so on. So we have these two cases. So let's first speak about this one. So the stinger pheromone, which is a complex bouquet, again, made of 40 molecules. When we say this, okay, a very important thing to keep in mind is that, of course, if you would like to reproduce the full effect of the pheromone, you need to have all the compounds there. But you might also need to consider that some of these components might be more important than others. So many times, and that was experiments done by many people, including people in the Rodham station actually, they tried to reduce the number of components to say, okay, I am able to reproduce the effect of the whole pheromone just by using this blend of 4 or 5. So, which is possible. Okay, so and in this case actually you have 4 components, but there is one of them which is extremely important, plays a dominant role and so on. It's not that this component is able to reproduce everything as the entire pheromone, but most of the things can be reproduced by this component, which is called isopentyl acetate, also called isoamyl acetate. I would just use this term, but I mean, it's abbreviated IPA, okay, which is the most important one. So it's released by this Koshevnikov gland here, and it's producing this typical, I mean, those who are beekeepers know that very well, this typical banana smell odor. You probably had that when you got stinging behavior and so on around you. And actually that's what it releases, actually. It triggers the coordinate attack, and actually probably the word attack is not the right one because these guys are not attacking, they are defending themselves. So that's the idea, it's the coordinate defense. So, and what is obvious actually is that by stinging you, they scent mark you with the pheromone. And this is why, I mean, you probably thought about that as being beekeepers, why the probability of getting a sting in the same place or very close to a place you already got one is very high. Just simply because you got a red cross there saying it's there. Okay, you don't see it, but it's there. Okay, and that's actually the effect of this isopentyl acetate or the whole pheromone, actually. So, and that's something different, actually, related to IPA, this component of the stinger pheromone that was discovered after the identification of this pheromone, which is something very funny, actually, very crazy, which is the fact that this IPA, this isopentyl acetate, induces something like an opioid-like analgesia in stinging bees. What do I mean by that? So you probably know about opioid analgesia. So you have opioid receptors in a nervous system, and actually why you take aspirin to reduce these, the pain and so on. It's because aspirin play on these opioid receptors and decrease the sensitivity to pain, okay? So that analgesia is exactly that. So it's play on the opioid receptor, and you take pills, and then your sensitivity to pain diminishes, you don't feel pain anymore. That's the idea. So, and the crazy idea here in this experiment was to show that of course, there is a lateral component of this pheromone, which is to act like an aspirin in attacking bees, to say it simply. So to activate or to produce opioid-like analgesia. So in this experiment, what they had is, well, I participate also in this work, but actually the main work was done by Norberto Valderrama. And he conceived a system in which he had a bee harness actually between two metal plates, okay? And the bee is making the bridge between these two metal plates. It cannot move. And he was doing that because he aimed at delivering very, very mild electric shocks to the bee, just to see, or just to trigger by doing that, the defensive response of the bees, which is the extension of the sting. Okay, so the bee is here, gets a mild shock, and then, pack, you see the sting extended. This is something that you can see here. The head of the bee is here. You are seeing frontally, and you see the shock, and you see the sting extension. So it's something that you can quantify very well. You see the sting here. And he aimed at actually asking the following question, which is doing the following experiment. He will expose bees, actually, again, to mineral oil. You know already that this has no smell, it's just a control. But he will expose a group of bees to a small dose of IPA, a higher dose, and an even higher dose of IPA. I call them dose 1, 2, 3, just to indicate 1 is low and 2 and 3 are higher, okay? And then he had these bees exposed to this mineral oil or IPA doses. He let them resting for a while, so like 15 to 30 minutes. So it's interesting because, I mean, when he provides the shocks, the pheromone is no longer there. So 15 minutes, 30 minutes have been elapsed since then. But he would like to know how do they respond to these different voltages that you can see here in terms of sting extension. And this is what was obtained. Okay, so just to go through this graph in a simple way, this graph here is the control group. And this control group shows what you expect, actually. So which means that they react a little bit to 2 volt here, but then you increase the voltage, they react a little bit more, 8 voltage, they react more, and so on. So this is the normal response. You get more voltage, you respond, you extend the sting even more, normal response. What happened now to the bees exposed to EPA? First dose, you have them here, these guys. You can see that response has been reduced. Second dose, you have the triangles here. Similar, but reduced again. Third dose, here. Okay? So these guys became something like insensitive to the shock just by being exposed to IPA. So the IPA is not only triggering the attack, the defense, and so on, but it's changing the sensitivity of the attackers to the noxious stimuli, to the aversive stimuli. So one nice metaphor that this paper provides actually is to speak about something like doped kamikaze warriors. So the idea is the following. You probably heard about the fact that the kamikaze warriors were given methamphetamines actually before the attack, so they were like dope, and they went in that way to crash planes actually against the enemy targets, whatever. So this is something like that. I mean, or the metaphor is going in this direction. So these guys have a reduced sensitivity to aversive stimuli when they go to attack if they have been exposed to the pheromone releasing this effect. So why we say that this opioid-like? Well, there's no time to show you that, but only briefly, because they use an antagonist of opioid receptors, which is called naloxone. The name is not important, but they use an antagonist, and they were able to revert the effect. So they showed that this was indeed based on kind of opioid system. Much later, we discovered the opioid receptor triggered by this situation. But that's another story. I think this is the interesting thing. It's just to see this aside interesting effect of this IPA, stinger pheromone component. So let's speak now about the other one. Remember I told you GARDS2, so the stinger pheromone and the mandibular gland pheromone, where I say we have just one molecule, 2-eptanone, okay? So this is 2-eptanone, which is released by the mandibular glands here in the workers, and it induces a repellent effect. It rejects intruders, so typically when you have this kind of mess of invaders, other bees from the colonies arriving there to rob or things like that, to rob a colony, then you use massive release of 2-eptanone to reject these intruders or things like that. Or like in this, you can see here, typically here a bee biting a moth larvae or things like that. There is also release. I mean, this movie was made to quantify it afterwards, release of 2-eptanone. And that's a typical situation where this molecule is released. But there are other functions that have been suggested and that stay under interrogation sign, I would say, which is some kind of very, very disturbing finding. If you would argue that 2-eptanone is basically a repellent pheromone for defense, in which category of bees would you expect to have more of this? I would say in the guards, okay? But that's not the case. The higher levels of 2-eptanone are found in the foragers. But foragers do not defend the hive. So one of the explanations and the hypothesis that has been provided to this kind of situation or this finding is that 2-eptanone might also play another role, which is to scent mark recently visit and empty flowers, so that when the bee goes back, and because it's a repellent substance, they would just go out without losing time in inspecting this flower. And this is why it would be so present in foragers. So that was one of the hypotheses that was provided. Indeed, it was shown that when bees visit an artificial patch of flowers and they go to empty flowers where they have been, they just put the head and immediately go out, like if they would perceive something repellent there. And then they connected to this artificial flower some air extraction that took the air within the artificial flower out. And so in this case, the bees accepted the visit flower without any repellency, just because probably you empty the scent mark. This is something that's still not proof, because what needs to be done is to take the air of this empty flower and put it in a chromatograph and finally prove that 2-eptanone is there or not. So it's still a question sign. But just to tell you that there are other hypotheses about 2-eptanone besides this repellency related to defense. Okay, so now we are coming to foragers. Okay, so we went, remember, we did queens, we did brood, we did guards. Now we're coming to foragers. And so these pheromones, I would like to mention those who are related to signaling in an appetitive context, so context related to food search. And obviously you all know the Nasonov gland pheromone, okay, which is extremely important for marking points or sources of interest. It's the Nasonov gland is a gland at the end of the abdomen. Bees typically expose it, and you have probably seen that actually at the hive entrance. It's possible to see that because they also mark the hive entrance with this pheromone to attract the incomers to the right place. Okay, and the Nasonov gland pheromone, again, it's a bouquet of different components. Okay, so in the case of honeybee foragers, we know that there are 7 components, of which one of them, geraniol, is the most important one. Again, like for the stinger pheromone, this is not to say that geraniol reproduces all effects of the entire gland perfectly. No, but it might reproduce a significant amount of these effects. Like the IPA, like isopentyl acetate, similar thing. It's the most important one, but do not forget that the pheromone is the whole thing, okay? So, and you have seen that obviously that release of the pheromone is, goes together with the flapping of the wings, so that they expand the action range of the pheromone. Okay, and it's very well known that this pheromone attracts to marketplaces, food sources, hive entrance, and so on. Okay, so well, this is a bee. Well, you don't see the wings because they are moving, but here's the Nasonov gland, and the wings are helping actually the channeling of the pheromone towards the receivers. And this is something new, and we're still in foragers, okay? But this is something new because this is something you might not be aware. It's very recent in a way, and unfortunately it has not been continued. So this is something that requires more investigation, but it's definitely there. So this is Harald Esch, you can see here, a very important bee investigator, a German investigator, but he was working at the University of Notre Dame for almost his entire life in USA. And he passed away in 2017, and this is why these experiments were not continued. Unfortunately, he was ill before. And, but he made a sensational discovery that is not well known so far, so it's a good occasion for you to hear about this, which is the discovery of a new forager pheromone, the foraging activating pheromone, which is released by the dancers during the dances. It was a kind of new discovery, and what Harald Esch and his student Corinna Thom did is to use this kind of extraction method, okay? It's called fiber grass chromatography. So you can have, for instance, something like this. You have a holder, a needle, a fiber coating. So you can actually put this on top of your individual and get a sample of the, say, air environment around this bee, which you might send to a chemical analyzer to get which kind of molecules are here around. Is it clear? Okay, so what he did is to use this kind of method that you can see here illustrated with this bee that is enclosed here, but in an observation hive. So he just inserted his analyzers, his device, into an observation hive. And before that, of course, he had trained some bees to go to a feeder. So he knows these are bees that were trained to a feeder. He marked them with a, say, whatever color, and here it's indicated red. But so he marked the bees, so he knows, for instance, that returning bees are marked in red. And now he looks at the dancers coming back from that feeder, but dancers who are red marked. So he knows, okay, these are the bees I mark at the feeder, and I know they are reporting actually about that feeder. And he put the device just on top of these dancing bees, these marked dancing bees, to characterize the environment around them, the chemical environment. And he found actually that these guys, the dancing bees, were consistently releasing a blend of 4 components, okay? The Z9 tricosine, tricosane, pentacosine, pentacosane, and so on, okay. Again, names are not important, but you know there is a blend of these 4 consistently released by dancing bees. They are dancing, remember they are reporting distance and direction. Not only that, they are releasing a pheromone, okay? And that's new. People didn't know that before. And then he said, okay, what's this doing? Why are they doing that? And so he did this very funny experiment in which he has actually an observation hive, and he has the molecules, and he will inject the blend of the 4 molecules into the observation hive, okay? He's heating that just to make the whole thing more volatile, okay? But he's injecting that into a hive. So here, actually, and he measured the activity of the hive as a result of this injection of the pheromone. So here's what you can see. This is the result that he obtained. So this is the normalized mean number of bees arriving at the feeder. So remember, the whole thing is related to a feeder he established in the field. So, and he looked, so he was at the feeder and he counted the number of bees arriving. So he had 2 treatments. One observation hive was treated with a solvent, which is just control. Nothing special should happen there. And then you see, of course, yeah, with the day, okay, of course, the blue bars, there is an increase in the number of bees arriving at the feeder. But what happens when you inject the pheromone? Okay, yellow bars, but this is the moment of injection. So until here you see solvent and pheromone, no differences. Now you inject the pheromone, look at what happens. Okay, yellow bars go up. So this experiment shows that this pheromone, this new pheromone, actually activates the departure of dance followers. So the idea is that the dancers are not only reporting the distance and direction of a food source, they are also stimulating, by means of this pheromone, the departure activity of the followers. So the followers are the ones who are exposed to this pheromone because they are in the immediate surrounding of the dancer. So this promotes even more their, let's say, willingness to leave the nets and go to the reported place, as you can see here. So this is something, to me, very nice, because it's not every day that you discover a pheromone, but needs more investigation. So what I would like to do now is to provide you something different, which is related to pheromone function, which is something that we were doing in the last years, which is to ask the question from a different perspective. So we know that pheromones play a role in intraspecific communication. This is what we have been seeing all the time. But here the question is different. Can they modulate behaviors that are not necessarily related with the information content? Can they act on something else? In other words, can pheromone change the responsiveness to reward or punishment? Can they affect motivation of the bees? Can they affect learning and memory? Okay, that's a question. So nobody asked this question because, as you can imagine, we say from the very beginning, pheromones trigger innate behavior. And now if we speak about learning and memory, we're not speaking about innate behavior. We're speaking about something totally different. So the question is here, if exposure to pheromone can change the way you respond to reward, to punishment, and can affect the way you learn and form memories. Okay, so, and that's a question we address in the last 3-4 years. And actually, the two important persons here are David Baracchi, was a postdoc in my lab, now is a young professor at the University of Florence, and Patrizia De Torre, who is also Italian, but a professor in another university in Paris. And so we analyzed if exposure to pheromones of different valence, by valence I mean either being repellent or being attractive, okay, changed the way in which individual bees respond to sucrose solution. Okay, so we have a typical test in bees, very practical, by the way, to test responsiveness to sucrose, which is this standard test in which you have your harness bees in little tubes like here, and you just touch the antennae, and you determine if they extend the proboscis or not. So here you don't give the reward. You just touch the antennae and see if they would like to have this reward or not. So this is no learning. This is an innate reaction to sugar, okay? They like it or not, they extend the proboscis. And actually, when you do that, you get typical responses like this. For instance, this is bees showing the proboscis extension response. So what you do is you provide different concentrations, 0.1%, which is nothing, it's totally dilute, you see, 0.3%, 1%, 3%, 10%, 30%. And obviously you see that when you have a population of bees, a group of bees, you see that the higher proportion of bees respond to 30%, and a low proportion respond to the dilute concentrations. You can quantify that not in terms of proportions in a population, but for every bee you can have a score. You can do it in the following way. You can have, say, bee number 1, 2, 3, 4, and then you have your concentrations here, 0.1%, 0.3%, and so on. And then you, for each bee, you measure what the bee did. So for instance, this guy responded, this is why there is a 1 here, only to 30%. So the score is 1. This one responded to 30%, 10%, and 3%, so it's 3. These responded to 4 concentrations, 4, and these responded just to 2 concentrations, 2. Okay, more selective bees, which respond only to the higher concentration, have lower scores. Bees responding to everything have higher scores. Okay, and so in this experiment done by David, by the way, he exposed bees to 2-heptanone You know already about it, so I won't go back to this. So it's the repellent pheromone released by guards, so a negative valence. And then he exposed bees also to geraniol, the positive one, the attractive one. And he wanted to see if exposure changes the way in which respond to sugar, but when the pheromone is no longer there. So typically, this is what he did. He put the little bees in the tube. Here you have a bee in a tube. Here's a filter paper with pheromone, and he exposed them for some minutes. It could be 15, could be even less. I mean, 5 minutes would be enough. And he has a group exposed to mineral oil, again the control, no odor. Another group exposed to geraniol and another group exposed to 2-eptanol. So after exposing them, he leaves the bees for 15 minutes, could be also 30 minutes, rest. There's no odor, there is air extraction, nothing. And then he will do the test of sucrose responsiveness. So there's no pheromone there anymore. And this is what he obtained. So remember, group exposed to oil, to geraniol, 2-eptanol, 15-minute rest, and then testing the different sucrose concentration. The control group exposed to oil, okay? As we saw, of course, they respond less to dilute concentrations, and the higher the concentration, the more responses. What happened to the group exposed to geraniol? You see, they respond more. Even to the more diluted concentrations, there is an increase in responses. And the group exposed to the negative pheromone, tac, goes down. It decreases. So the same if you look at the scores. Remember, we quantified for every bee the response. It shows that when you were exposed to oil, you respond in average to 3 concentrations, score 3. But when you were exposed to geraniol, you increase and respond in average to 4 concentration. And when you were exposed to negative pheromone, 2-eptanol, you respond to just 1 concentration in average, okay? You see there's a lot of bees here, so that is a very robust effect. So the appetitive pheromone increases sucrose responsiveness. Bees become more sensitive to sugar and respond even to the dilute concentrations. The aversive pheromone, actually, they made the bees less sensitive and they respond less to all concentrations. So then the question came, okay, so pheromones are able to change the sensitivity even when they are there, when they are no longer there. So it means that they might also change the way the bees learn and make memories. Because, you know, for learning, you need to be sensitive to reward. And this is an experiment he did. That's the question, do pheromones modify learning and memory? And he used the protocol I explained yesterday, which is the olfactory conditioning of the proboscis extension. But I realized that some people might not have attended the lecture I gave yesterday, so I will show this again. So here it is. So this is what I explained yesterday. This is the way you can study olfactory learning in the laboratory with bees. You have bees harnessed in a small metal tube, like you can see here. Only the head is protruding from the tube. And this is a machine which is sending a constant clean air towards the head of the bee. And you have some cartridge here in which you can put some odorant molecules. And then you can make the air pass through the cartridge, so you can send to the bee a specific odor stimulation that you control to the millisecond level, so the duration of the stimulation. So the bee will be exposed to an odorant stimulation. The odorants that we use have no biological meaning to the bee, so the bees will not respond to this odor. And immediately after that, you see, well, this is the bee, this is the machine, and immediately after that, after sending the odor, the experimenter will touch the antennae with sugar solution. You see here a toothpick, which has been soaked in sucrose solution, and touch the antennae to release the extension of the proboscis. But this time, the experimenter will allow the bee to lick the sucrose solution. So in this way, as we say yesterday, the bee will normally associate, every time the odor arrives, I get sugar reward. And then, if everything goes well, the bee should learn odor means sugar, okay? And so, this is what we see here. So this is the first part. So you will see a red square here showing the moment in which the odor arrives for the first time. You see no reaction of the bee in particular, but now you give the sugar, you see the extension of the proboscis. You see, this is a very conspicuous reaction that you can measure very well. And after doing that, okay, normally you have created an association between the neutral odor, or the odor that was originally neutral, and sugar reward. And this is what you see here. Now you will see the odor arriving, and there's no sugar. But Nevertheless, the bee extends the proboscis to the odor because odor means sugar. Okay? This is something, as we saw yesterday, that takes place in very few trials. 2 or 3 are enough for all the bees having learned and leads to long-term memories that you can observe several days later. I will not go back because to this point we saw it yesterday. So what did David do? He did an experiment which was a little bit more complicated than that, in which he had 2 odors. Well, essentially it's simple. It's one odor rewarded and the other not. So the bee just had to learn to make the difference, okay? You get odor A rewarded, you extend the proboscis. But every time you get odor B, because it's not rewarded, refrain from doing that, okay? It's called differential conditioning, simply because the bee has to learn to make the difference between the 2 odors. One is rewarded, the other not. So this is shown here. This is odor which is rewarded, which is indicated by the red. So red means sugar, and here means nothing, no sugar. So 2 odors. Okay, one odor associated with sugar, the other odor not rewarded. And okay, and this is what he did. He exposed bees. One group was exposed to geraniol. Remember, that's the positive pheromone, the appetitive one. One group was exposed to 2-heptanone. Remember, that's the negative one. Okay, and another group also to a control group exposed to mineral oil. And not only he did the learning, but he did memory tests, asking the bees if they would remember what they learned 2 hours, 24 hours, and even 3 days later. Okay? So you can see the pheromone is no longer there. Okay? The pheromone was just in the very first minutes of exposure, then a lot of time goes away, and then you start doing your training, and then your test, and so on. And this is what he obtained. So here you see 3 groups, one exposed to oil control, one group exposed to geraniol, one group exposed to 2-heptanone, rest, then you do the training, and then you can test for memory 2 hours later, 24 hours, or 72 hours later. Okay, this is what he obtained. So I'd like you first to concentrate on this gray curve here, okay? So this is the group exposed to mineral oil, the control group. And you can see that these guys learn to respond to the rewarded odor, as you would expect. You know that they learn very well. And here's the response to the non-rewarded odor. You can see these gray squares here too. So you can see they learn to make the difference. They respond to the rewarded odor, and they inhibit the response to the non-rewarded odor. Good. What happened to the bees exposed to geraniol, the positive, the stimulating pheromone? Look at the learning performance. It's the blue triangles here. It's even better than the control. This is significant. For the non-rewarded odor stays at the same level because this is the bottom level. You cannot go lower than that. What happens to the bees exposed to the negative pheromone? Look here. It goes down. It's worse than the control. So for learning, it seems that when you're exposed to the positive pheromone, that tunes you to learn better. And where you're exposed to the negative pheromone, that actually impairs your learning. What happens in terms of memory? Here you have the result. 2 hours later, okay, control group, they have obviously a memory of what they have learned, but look at the memory of the bees that were exposed to geraniol. Back, higher. And the bees exposed to 2-heptanone, lower. 24 hours later, same result. 72 hours later, same result. Okay? So what we are showing here is that when you are pre-exposed to geraniol, you have improved learning and memory capacities. And if you're exposed to the aversive pheromone, you had impaired learning and memory capacity. And that makes sense if you think well. Because if you're exposed to an appetitive pheromone, a positive pheromone related to foraging, then you should learn and memorize better about foraging events. On the contrary, if you're exposed to an alarm pheromone, it's like, I mean, why should you learn better about appetitive events? You should actually focus on other things, defense or things like that. So you should actually detract from this kind of situation, and this is why you have this learning impaired. The conclusion is that depending on their balance, pheromones might increase or decrease learning and appetitive memory. So the missing experiment here, but we have done, it's only that I didn't include because it would be too much, is we use now the 2-heptanone, and we did now not an appetitive learning, not a learning in which they learn other sugar, but a learning which is aversive, which is a learning in which they learn other shock. And as you could imagine, in this case, it was the opposite result. When you were exposed to 2-heptanone, then your aversive learning was much better. But if you were exposed to geraniol, which has nothing to do with learning about bad events, then your aversive learning was worse. Okay, so it's the same but in the other direction. So the main result is that the time of passage prepares the bees for appropriate and more effective learning and memorization. So, well, this is the result you have seen already, the effect of geraniol and the effect of 2-heptanone, actually. I'm not repeating myself here, but the main message is that the pheromones improve the behavioral responses even when the perceived pheromones are no longer there. Keep in mind that, of course, the pheromones were not there when the bees were trained, were learning, were subjected to the memory test. There's nothing there, and nevertheless, the animals were tuned in the long delay, in the long term, to change the learning and memory capacities. So this is something that was very exciting to us, and one of the questions we had, and you might have actually, is this something unique to bees? And the answer is no, okay? This is a very recent paper, actually, or relatively recent, that was reported by the group of Richard Axel. And Richard Axel is Nobel Prize winner, actually, because of studies on human and mammalian olfaction. And they focus on mice, actually. And in mice, a pheromone was characterized, which was called darsine, okay? Darsine, because they use this name because they name it after Mr. Fitzwilliam Darcy, actually, which is the attractive hero in Pride and Prejudice, Jane Austen's famous book. And so it's called darsine, actually, and this is a substance in the male urine which attracts females to the males, okay? So, well, like this cartoon, and you can see, okay, I have to switch this pheromone off, I need a good night's sleep. You can see here. And actually, they published this article, actually, in Nature, where they say, yeah, surprisingly, this pheromone is not only inducing this attraction of the females towards the males, but it also improves the female spatial learning. So the females are the receivers of the pheromone, and not only are they attracted, but it changes also the capacity to learn about spatial problems and other problems. So, they conclude that something related to what I just said is that, of course, it induces a state of sexual drive, which is the role of the pheromone, but it also changes the probability of producing behaviors that somehow improve your tuning to the situation, okay? Not only just sexual attraction, like, for instance, spatial learning or other forms of learning. So this seems to be another property that has been recently discovered about pheromones. So, and keep in mind the following: pheromones, of course, they are chemical messengers, and of course their primary role is to convey a specific innate message about something like sexual attraction, territory, defense, and so on. But now the finding is that they might have aside roles or aside functions, lateral functions, like modulating the capacity to learn and memorize in order to improve the performance you're supposed to do. Okay, that's a new thing. So I'm finishing here. So what I try to do today is, first of all, to show you that insects, humble insects, are at the origin of the discovery and definition of the concept of pheromone, the case of Bombyx mori. So pheromones, everyone speaks about pheromones, of course, but everything started with a little silk moth. And thanks to Schneider and Buchanan, we got that. So pheromones are present in most animal species. Of course, as I say, they are chemical messengers. They are an essential communication channel, and they convey messages of different content between members of the same species. They are one of the pillars of sociality in bees that we know, we saw. And they are produced by different members of the society. We saw cases like queen pheromones, brood pheromones, workers, and different categories. We saw guards, we saw foragers, and they have multiple effects which ensure somehow the proper functioning of the society. And the final point is this one. Pheromones are not exclusively communication agents, of course they are, but they also what I would call cognitive agents. So they play also on the cognitive capacities of the receivers because they affect them. They modulate learning and memory, and somehow they prepare the bees to learn and memorize in an appropriate way, in a persistent way. Remember the test 2 hours, 24 hours, 72 hours later, in a persistent way about situations that need to be tuned to provide an appropriate response. So that's all. These are the people I mentioned. David did many of the works in our team, and these are other collaborators that, who participated in this research in the part that refers to what we did.
There is one area of colony activity you haven't mentioned, and that is when the queen is going out on her mating flights.
Yeah.
And I presume that when she comes back to the reception committee on the hive, they are assessing something on her to see whether she's been adequately mated, and I presume that must be pheromonal.
Yes. So concerning this situation, which is related to the nuptial flight, something interesting is that the chemical composition of some of these pheromones in the queen changes after reproduction. So the virgin queens might have some components in the queen mandibular pheromone or in the tergal pheromones, but this component changes after reproduction. So when they go back, I mean, in the time, I mean, there will be a significant change, and some of these effects will be accentuated. So you're right. So this event you mentioned, which is the nuptial flight, will have a significant impact in the pheromone production. Yeah, we could have mentioned that. You're right. Thank you for calling my attention to that.
As beekeepers, we use smoke to control a colony where we're inspecting it. Now, smoke, the smell of smoke lasts and persists for days and days. Has any research ever been done on how long the interruption persists in a colony after using smoke as a result of pheromones?
Not to my knowledge. The only research I know was done in the 80s, but in a different context, which is the impact of some treatments to induce amnesia in bees related to what they have learned. And in this particular study, I remember well, they use, for instance, cold treatment, and that was a very powerful amnesia agent. So after recovering from the cold shock, they didn't remember very well. And they did also smoke, and smoke had similar effects. So it's not answering your question. Unfortunately, I don't have the answer. But at least what I can tell is that if they have learned something, the smoke treatment might somehow impair this memory of what they had.
So conversely, is there not an opportunity, therefore, to have a pheromone-based control agent that beekeepers use, which has a much less impactful effect after we've finished the inspection?
Yeah, well, there is something I can tell, but I have no idea if it will work, because you need commercial assays and so experiments and so on. And as I tried to explain yesterday, we try to contribute knowledge, but this is not the kind of applied research we do. But one of the papers we published a couple of years ago was a crazy story, actually, inspired by someone who mentioned to me, you know, actually, that there is a kind of relieving effect in humans when we smell the odor of cut grass. You know that, okay? There is like, oh, good feeling and so on. And say, would there be something like that in bees? And of course, we did some experiments, and we end up identifying two molecules. Linalol is one, the other is a phenylethanol or something like that. And these were, and actually we play with the impact of these molecules on stinging behavior. So we had a special device where we had, say, 2 or 3 little bees in a petri dish, and there was a rotating plume which actually was bothering them, and they typically go onto the plume and attack and sting them. But when we put into this chamber these two components, either one or the other, they were like calm and relaxed. And we published that. So this is a published work. It's what's the journal? I think it's Nature Communications. And we showed that actually there are some molecules which innately somehow diminish the aggression levels, which doesn't mean that I have the solution to apply that at the colony scale, of course, but at least at that level it works. And even more, so we showed that when you take newborn bees, naive bees, put them in these little tubes, okay, and blow the odor, they extend the proboscis to the odor. Remember that actually they extend the proboscis to sugar contact. Here there's no sugar, but the odors themselves, these two odors, had a kind of innate appetitive value. We did it with bees in Australia and bees in France, and the same result was found. So these two odors seem to have a kind of particular value which put them in an appetitive mood and then detract them from attacks or things like that. That's the only thing I can say. But okay. Far away from any commercial application.
We'll just take one more down at the front here.
Yeah. Hi, David McClellan from Exeter again. You're looking at all of those bouquets of chemicals. There's an awful lot there. You're doing tests with sucrose, etc. But again, this comes back to the diet of the bee, what Roger was saying yesterday. You need a range of fodder to provide that. So has there been any research into the foods that then would provide these pheromones? And is that part of the problem with colony collapse if they don't have it?
Yeah, I mean, this is it's a fascinating question actually, which is how the nutritional levels or nutritional components may change all the things we are seeing here. And you might remember, oh well, by the way, you can see her here. I mentioned Gabriela actually, who is here, who is the one actually studying all these mechanisms controlling appetitive motivation, regulation of feeding decisions, and so on. And she's very concerned about these questions. And one of the things she found is that actually there are some molecules, I can tell you the names. One is dopamine, which actually is activated by social needs. Okay, so and that is related to what we call a wanting system. So when you want something, even in humans, dopamine levels increase and are released by specific circuits. And we found that bees have exactly the same. What is the main difference? Is your wanting system is activated by your individual hunger and needs. Their wanting system is activated by social needs, not by their individual needs. So that is a level of the supraorganism, you see? It's not the individual. But I mean, and of course it depends on which nutrition do you have and things like that. And there is another molecule, it's a neuropeptide. These all things are published, so you can eventually find those papers called the neuropeptide F, okay, that's name, just, and which plays also a significant role depending on nutritional levels also too. Once you decide where to go and that you're going there, dopamine based, and then you reach the place, it's also actually modulating your willingness to eat more or less. Well, now remember, beta-ocimene was doing that, okay? So the question is beta-ocimene changing the levels of these molecules internally to make them change their behavior in this way? That's exactly what we are trying to figure out now. So we have no results, but you see, we're playing on that right now, okay?